Have you ever asked, what is biotechnology? It sounds interesting, but I just have no idea what it is exactly. Well, we tend to think of biotechnology as a relatively new field of science, but the word biotechnology was first used in 1919 by a Hungarian engineer. He coined the term biotechnology to describe using another living organism for one's own benefit. Given the date of this definition, biotechnology is not a new idea. In fact, it dates all the way back to the beginning of civilization. From the beginning of time, agriculture has concentrated on improving food production. The earliest and most familiar bioprocess is microbial fermentation. Fermentation is a metabolic process where the byproducts of glucose metabolism, carbon dioxide, and lactic acid can be used to make useful products such as bread, wine, yogurt, and cheese. For thousands of years, humans have used an agricultural biotechnology application called selective breeding. Selective breeding is the process of breeding plants and animals for desirable genetic traits, such as high crop yields, resistance to disease, and high growth rate. It's common sense to know that if you breed a male and female with a desirable trait, the offspring will all carry that trait. However, until a few hundred years ago, no one understood the biology behind this observation. Today, the biotechnology of marker-assisted selection, or MAS, allows breeders to use biochemical and genetic markers linked to traits of interest to identify superior plants and animals for breeding. In the 1800s, Gregor Mendel began studying inheritance patterns of the common garden pea. Mendel's observations led him to believe that some traits were dominant and would always be expressed in the offspring, while other traits were recessive and would only be expressed if both parents contributed a recessive allele. Mendel's work went largely unnoticed until the early 1900s and has since been considered the foundation of population genetics. It is important to note that during this time period, no one understood that DNA was the mechanism of inheritance. Today, the structure of DNA is known, allowing for countless biotechnology applications. In the late 18th century, Edward Jenner demonstrated that people injected with the live cowpox virus would become immune to smallpox. How could that have possibly worked? Well, to understand how it works, you need to know a bit about the immune system. A pathogen is a bacteria or virus that has the potential to cause disease in the host it invades. Your body reacts to these pathogens by stimulating white blood cells to produce antibodies. These antibodies attach to the pathogens and mark them for destruction. After clearing the infection, some of the white blood cells become memory cells. So if your body ever encounters the same pathogens again, the memory cells can quickly produce large amounts of the antibody and attack the invader. So how did Jenner's vaccination work to protect people from smallpox? The cowpox virus is very similar to the smallpox virus. So while the cowpox injection did not cause people to get sick, it did stimulate the immune response and produce memory cells that would recognize the smallpox virus if the person ever came in contact with it. Therefore, the injected people had immunity to the smallpox virus without ever becoming infected. Today, more than 325 million people worldwide have been helped by biotechnology-derived drugs and vaccines. Another major advancement in the pharmaceutical industry was the discovery of penicillin. In 1928, Alexander Fleming noticed, by accident, that colonies of the bacterium Staphylococcus aureus could be destroyed by the mold Penicillium notatum, providing that there was an antibacterial agent there in principle. Fleming, a typically inquisitive scientist, investigated the cause of this phenomenon. Antibiotics work in two ways: by directly killing the microorganism or by inhibiting its replication. Antibiotics only work for bacterial infections. Antibiotics do not work on viruses because viruses are not alive. Since Fleming's discovery, many new antibiotics have been produced by the pharmaceutical industry, but there is still only a limited number of them. It is important to maintain the potency of antibiotics by controlling their overuse and ensuring that patients finish their antibiotic prescriptions. When antibiotics are not used correctly, the bacteria can mutate and begin to build a resistance to the antibiotic. Accordingly, when people think of modern biotechnology, gene engineering and recombinant organisms take center stage. Biotechnology was revolutionized when scientists first learned how to isolate and clone genes, making it then possible to insert the cloned genes into different organisms. For example, scientists have isolated a gene from a jellyfish called the GFP gene. This gene expresses a protein that will fluoresce green under a UV light. Thus, the gene is called the green fluorescent protein. Scientists have been able to clone the GFP gene into many other organisms, including the fish in this picture. And when the GFP gene is expressed, these organisms glow green under UV light. One thing is certain, regardless of where your interests lie, there is an application for biotechnology. Biotechnology applications are used in the fields of agriculture, bioremediation, food processing, textile production, forensics, archaeology, and medicine. Biotechnology encompasses a number of controversial topics that you have heard on the news, such as cloning, the use of embryonic stem cells, animal testing and research, genetically modified food, and creating designer babies. With the advances of biotechnology, we're on a great journey to solving many of science's mysteries. The answers lie in our ability to better understand biological processes and how to use the biotechnology applications to derive solutions.